Welcome back, everyone. So we're continuing on with Chapter 7, Patterns of Inheritance. This also continues in Chapter 11 of the Campbell Biology textbook. So we're getting into laws of inheritance. So this continues on with what we learned in Chapter 7.1 with Mendel's experiments. We're taking it a step further and we're applying what we know now about genes and chromosomes to what Mendel learned from his experimentation. So we're kind of putting it all together and into this laws of inheritance and understanding how we inherit things from our parents with our genes. So by the end of this video, you should be able to explain the relationship between genotypes and phenotypes in dominant and recessive gene systems. You should be able to use a Punnett square to calculate the expected proportions of genotypes and phenotypes in a monohybrid cross. You should also be able to explain Mendel's law of segregation and independent assortment in terms of genetics and the events of meiosis. And then explain the purpose of and methods of a test cross. So we're going to be getting into basically a lot of new terminology, as well as we're taking information from last week with meiosis, and we're applying it to genetics. Okay, so it's basically building. All this information has been kind of building, so we're taking it all and bringing it together this week. Okay, so in the previous video we talked about Mendel's experiments, and remember how in his experimentation he figured out that each parent pea plant had two discrete copies of a characteristic. And those copies were passed on to the offspring, so that the offspring each had one copy from each parent. Okay, so for example, in this example with the the pea plant, the coloration of the flower came from both parents, right? So this F1 generation got a copy of the coloration from each parent, got a copy from this parent saying that it was going to have violet flowers, and got a copy from this parent that said it was going to have white flowers. However, so now it has two copies. It has a violet color copy and a white flower copy. But the violet color masks the effects of the white color, and so that's why it shows the violet color instead of the white color. Okay, so later on, now this was back in the 1860s when he figured this out. But now we know that those copies, those are genes, right? And those genes are carried on homologous chromosomes. And we each get one copy of chromosomes from our parents, right? We got one of those homologous chromosomes from our, mom, and the other one came from our dad. And each of these copies carry the genes. So we have two copies of every gene, okay? We got one from mom and one from dad. And if these genes code for a certain characteristic, in this case, flower color. And so we have two copies of every gene. So we have two copies of the flower color gene for this pea plant. And if the two copies are the same, okay, they're gonna, for example, this one, the copies are the same, they're violet color. This one's the same, it's white color. But this one, it's different, right? Okay, so this one, it has one violet color gene and one white color gene. So if we have two copies that are slightly different from each other, they're called alleles. Okay, so alleles are variants of the same gene. Okay, they still code for a flower color, it's just the variation is slightly different. There was some mutation that produced a white flower instead of a purple flower a long time ago. And so a variant of a gene is called an allele. Okay, so the F1 generation in this case has two different alleles, has a violet allele and a white allele, okay? Whereas in this violet flower plant, parent plant, it has two violet alleles. This white flower plant has two white alleles, okay? So we're going to start using some new terminology to call these different genes and color and variants of genes and things like that, okay? We're going to also use some new terminology to determine like the alleles and the when we have two of the same versus two different alleles and so forth. So we're going to start off with an Amoeba Sisters video. This Amoeba Sisters video goes into alleles and genes, as well as some additional terminology that we're going to use throughout this lecture video. So it'll give us kind of a good overview of what we're going to be learning about. Okay, so let's go ahead and watch that video.
I don't remember which grade it was where I learned something about my taste buds that can never be unlearned, but the event in the lesson with genetics has stuck with me forever. For you see, I learned that my taste buds cannot taste PTC. PTC stands for this. We'll stick with PTC. And it's a chemical that can be sold on these paper strips. It can be purchased under the name PTC paper, and it's popular in genetic classes because it has this fascinating quality. Some can put it on their tongue and immediately say, yuck, this is bitter. And some people, when they place the paper on their tongue, taste absolutely nothing. Well, unless you consider the paper. Does paper have a taste in itself? That's a debatable question, but the point is some people can taste PTC and some people cannot taste PTC. And I was really disappointed because I remember that I was the only one there that could not taste it. So there was everyone getting this amazing science experience and I couldn't taste a thing. Well, there may have been more than just me that couldn't taste it in the classroom that day, but they didn't seem as concerned by the fear of missing out of the PTC paper as I was. I remember someone trying to make me feel better, saying, oh, but it tastes bitter, you're actually lucky. Then they tried to describe what it tasted like to me, but it's not the same. I guess I'll never know for myself what it would have tasted like. Of course, the reason PTC paper is used in genetic classes is because the trait of being able or not being able to taste PTC is based on genetics. A reminder from our Intro to Heredity unit that genes are portions of DNA, and they have the ability to code for a characteristic, a trait, like being able to taste or not to taste PTC. Now, we do want to point out that many traits are actually coded for by interactions of more than one gene, like eye color, which is quite complex and determined by interactions of many genes together. In fact, the ability to taste PTC or not may involve some other gene interactions. There's even different ranges for how bitter the chemical may taste because there may be more kinds of alleles than we'll mention. More about that later. But since we do know that the ability to taste PTC or not taste PTC is at least heavily impacted by a specific gene, it does make it powerful for genetic classes. One thing I have found so interesting is that my parents can both taste PTC. So why can't I? Recall that humans have 46 chromosomes. Chromosomes are made up of DNA and protein. It's a condensed unit of DNA. My whole genetic code is represented by these chromosomes. You inherit 23 chromosomes from your mother and 23 chromosomes from your father. Here's all 46 of them right here. As you can see, there are 23 chromosome pairs. Each pair has one chromosome from one parent and one chromosome from my other parent. If we focus on one of these pairs of chromosomes where the PTC taste sensitivity gene may be found, we can see an area where the PTC taste sensitivity gene could be. Let's assume this is the locus where the PTC taste sensitivity gene is found. See how it is pointing to a specific area here? That's because it's on an area of the chromosomes that refer to a specific gene that codes for a trait. Now, remember how this chromosome is from mom. This one is from dad. Each parent contributes an allele, which is a variant of a gene. An allele is a variety of a gene, a form of a gene. The alleles could be the same form of the gene or different forms of the gene, But regardless, in this case, they're forms of the gene involved with PTC taste sensitivity. So if PTC taste sensitivity is being used as a one gene trait example, and as we mentioned, it may not be that simple, then your DNA code has a gene related to PTC taste sensitivity. Together, the two alleles you inherit, the forms of that gene, determine the trait of tasting PTC or the trait of not tasting PTC. That gene is involved with coding for taste receptors on your tongue, and the receptors you have can make a difference for whether you taste PTC or not. The alleles are typically represented by letters. Since this is all about tasting, let's use the letter T. But wait, it matters whether I represent it as a capital or lowercase letter. If I use a capital letter to represent an allele, it means that it's a dominant allele. If one or both of the alleles you inherited for a trait are dominant, then it will be expressed. More about that later. If I use a lowercase letter to represent an allele, that means it's a recessive allele. Recessive alleles are typically not expressed unless there is no dominant allele present. Now remember that you have two allele copies, so the combination you could have here could be big T, big T, big T, or little t, little t. These are called genotypes, your genetic makeup. Genotypes can help determine a phenotype, which is a physical characteristic. You'll notice when writing genotypes, I put the capital letters first, if it contains a capital letter. That's not because the order matters, it's a formatting formality that capitals are written first. It turns out that being able to taste PTC is a dominant trait. That means the phenotype, which is a PTC taster, is due to a genotype that includes at least one dominant allele. So which genotypes can taste PTC then? Well, big T can. Both of those alleles are dominant. So can big T, little t, because remember, it only takes the presence of one dominant allele. In fact, the only genotype in this simplified example to not be able to taste PTC would be little t. So obviously that is what I am. I am the little t genotype, which results in my non-taster phenotype. But my parents can taste PTC. So what genotypes would they have to be? Well, if they were both big T, that wouldn't be possible. If one was big T, and one was big T, that still wouldn't be possible. Remember, you have to get an allele, a form of a gene, from each parent. If my parents do taste PTC and I do not, then my parents have the genotype big T, and their phenotype is PTC taster. Punnett squares can be used to determine the probabilities of offspring having certain genotypes, which then can be used to determine their phenotypes. But Punnett squares are for another Amoeba Sisters video. Before we end, one more thing to mention. In this example, the dominant trait of being able to taste PTC is more common than the recessive trait of not being able to taste PTC. And one could jump to an assumption that dominant traits are more common, especially since it only takes the presence of one dominant allele to show up in the phenotype, at least in Mendelian inheritance. But the dominant trait is not always more common in a population, because it's possible that the dominant allele itself is more rare. That can be the case with some forms of polydactyly, that is, being born with extra fingers. Some forms of polydactyly can be a dominant trait caused by the presence of at least one dominant allele. However, the dominant allele may not be as common in the population, and the condition of having extra fingers is generally rare. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so again, that was a good introduction into some of the vocabulary and terminology that we're going to be working with for the next couple of videos. So in the video, the Amoeba Sisters mentioned like the phenotype versus the genotype and homozygous versus heterozygous. So we're going to go into those terms as well. So when we're looking at an actual trait, something that is observable, we call it a phenotype. So for example, in the video, the ability to taste something, that's the phenotype. So whether she could taste PTC or not, that's the phenotype. Okay, so the ability to taste was the phenotype. So she couldn't taste the bitter taste, but then the rest of the class could. So she was a non-taster. So her phenotype is non-taster, but the rest of the class are tasters of PTC. So they're PTC tasters. So that would be their phenotypes. Whereas a genotype is the underlying genetic makeup. So for her, she has 2 recessive alleles. So she basically, since she cannot taste the bitter taste gene, remember how they used the lowercase letters to denote a recessive allele and then the uppercase letters to denote a dominant allele. So in her case, she lacks the dominant allele that allows the tasting of the PTC. So she has 2 recessive alleles, and so they denoted that by lowercase t, lowercase t. So both of her alleles are recessive. And so this would be her genotype, her genetic makeup, her underlying genetic makeup. So she got one of her little t's from her mom and the other little t from her dad. Okay? And so since both of them are the same, we call that homozygous. Okay, so think homo means the same. So homozygous means having 2 identical alleles for a given gene. And since both of these mean recessive, you can call it homozygous recessive. Okay, so she, in this case, is homozygous recessive for PTC or bitter taste gene. But her parents, remember her parents, they could taste the bitter taste. So they have a dominant allele, right? So her mom has a dominant allele, her dad has a dominant allele. But they're her parents, so they gave her recessive allele, so they must have a recessive allele. Okay, so each parent must have given her a recessive allele for her to have the non-taster gene, right? Okay, so that means that her parents have 1 dominant allele and 1 recessive allele, so they are considered heterozygous. So hetero means different, so they have 2 different alleles for a given gene. Okay, so their 2 alleles are different. They are expressing the dominant phenotype, meaning that they taste the bitter taste, but they carry the recessive allele which they passed on to their daughter. All right, so again, phenotype is the physical expression of the trait. The genotype, think genetic makeup, what the genes say. All right, so here's just kind of an example showing that, going back to Mendelian genetics. So we have the phenotype is the physical traits. You can see here we have a yellow pea and a green pea. So we're talking about the characteristic of the color of the seed. Okay, so we have yellow and we have green. Okay, if we just look at the phenotype, we can see that the parent generation, okay, they're yellow and green, and then we allowed them to, hybridize, and then we took the offspring, and we looked at the offspring, and all of them, the flowers of the offspring or the plants of the offspring produced yellow peas. Okay, so 100% yellow. All right, so all of them produced yellow seeds. But then we allowed them to self-fertilize, and then looked at those seeds that were produced, and about 75% of them produced yellow, 25% produced green. Okay, so that's the phenotype. But if we look at the genetic makeup, we can see that the yellow, its genetic makeup is homozygous dominant. Okay, it has 2 dominant alleles, and remember dominant, we denote dominant with an uppercase letter. So in this case, we just chose the letter Y for yellow. So uppercase Y, uppercase Y. And then the other color is the recessive. So green is recessive, so we just denote that with lowercase y, okay? So then if we hybridize them, cross-fertilization takes place. One of the big Y's will end up in the offspring, the other little y ends up in the offspring, and so here is the genotype of the F1 generation. Okay. And then of the F1 generation, if we did a Punnett square, we could write out that Punnett square like I did earlier in that other video. So if these gametes, so if this was one parent, half of the gametes that were produced through meiosis would be a big Y, the other half would be a little y. So big Y, the other half would be a little y, okay? In meiosis, the other parent would have, half of its gametes would have a big Y in it, the other half would have a little y. So then if I did this Punnett square, this Y I'd bring down, this Y I would bring down, this Y I would bring across, and this Y I would bring across. And this is the result of the Punnett square. So I would have about 25%, a 1 out of 4 chance of getting homozygous dominant. I'd have a 50% chance of getting heterozygous and a 25% chance of getting homozygous recessive. Okay, so you can see where we're getting all these numbers from just by doing that Punnett square. Okay, it's a lot easier than just saying, hey, I went from this to that. How did I get that? Okay, so by doing a Punnett square, you can kind of see where we got it. And we'll be practicing Punnett squares in a little bit. Okay, so for now, okay, just realize that if you have two of the same alleles in your genotype, it would be homozygous. And if they're two uppercase letters, it would be dominant. If they're two lowercase letters, it would be recessive. Okay, if they're two different alleles, it would be heterozygous. Now, when Mendel was doing all of his work with the garden peas, he was discovering a lot of different things, and his experimentation allowed us to come up with some scientific theories, which still hold today. And so, one of the laws of genetics that still holds today is something called the law of dominance. So Mendel's law of dominance states that in a heterozygote, one trait will conceal the presence of another trait for the same characteristic. So that means if we have a heterozygote, big Y, little y, the big Y, the dominant allele, will mask the effects of the recessive allele. So in this case, whatever the dominant allele is will be the phenotype that's being expressed, okay? The little y will not show up in the phenotype, okay? So this one is showing the pea color. So if we have a heterozygous for pea color, it will show the yellow coloration for the pea. It won't show the green, okay? The only way that we will see the recessive phenotype is if we have only recessive alleles, okay? If we have a dominant allele at all in the genotype, it will show the dominant phenotype, okay? So the only way we can show the recessive phenotype is if we have two recessive alleles, okay, in the genotype. So here's an example. Albinism. So albinism is a recessive trait where you have to have 2 recessive alleles in order to exhibit albinism. So this young boy, he has albinism, so that basically means the lack of melanin in your skin. So melanin is a pigment in your skin, and so if you don't produce that pigment, then you're basically lack that pigmentation. And so people with albinism, they have to be very, very careful when being outside, because they're very prone to skin cancer and things like that can damage their skin because they don't have that natural melanin. So we know his phenotype, it has to be homozygous recessive. Okay. We also know his parents' genotypes, they must be heterozygous if they exhibit the dominant allele of producing melanin in their skin. Okay? So that's the law of dominance. So the heterozygote must possess the dominant allele that masks the effects of the recessive. So that means that if 2 heterozygous people or individuals mate, they could produce an offspring that is homozygous recessive for that given trait. And there's about a 25% chance. Okay. So we're going to watch a video, an Amoeba Sisters video, on basically on monohybrid crosses. So this will give us a good introduction into like Punnett squares and how to set up these Punnett squares, and we're starting off slow, so we're just crossing individuals that differ in one variant, or one trait.
So I have a confession to make. For my classroom, I always wanted a hairless guinea pig. They sell them at some pet stores, but they can be really hard to find. And while I doubt they do so well in the wild, their genetics are fascinating. And so are guinea pigs. We spend quite some time on LOL cats, but we'd spend even more time if someone would make a funny guinea pig site. Most guinea pigs have hair. This is because of their DNA. DNA makes up genes, and the guinea pigs received genes from both their mother and their father. An allele is a form of a gene, and often represented by a letter. In this case, we'll use the letter H for hair. A hairless guinea pig has 2 recessive alleles for the trait of having or not having hair. A recessive allele is usually represented by a lowercase letter, or allele. By recessive, it means that the allele will not usually show up. The only way it will show up is if there is no dominant allele present. A dominant allele is represented by a capital letter, and it's an allele that will show up. Think of it as dominating. So a hairless guinea pig does not have a dominant allele for hair, and that's why the recessive trait, not having hair, shows up. A hairless guinea pig's genotype, that's the genetic makeup of an organism, can be represented as a lowercase h. A guinea pig that does have hair can be represented as a big H, or a big H. See, it only takes one dominant allele, a capital H, for a trait to show up. That dominating allele means that the recessive allele is hidden. Now, we're not quite done with the vocab here. A genotype of big H, or little h, is considered homozygous. The root in this word means same, and they are the same case. Big H, those are both capital letters, and little h, those are both lowercase letters. So big H is called homozygous dominant because of the capitals, and little h is homozygous recessive because of the lowercase. A genotype of big H is considered heterozygous. The root in this word means different because their letters are different cases. One is a capital and one is a lowercase. If I were to show you a guinea pig with hair, would you know its genotype? Well, you would know it's not little h, because if it was little h, then it would be hairless. But remember, a guinea pig with hair, it could be big H, or big H little h? We don't really know. We could do a test cross to eventually determine this, but you can't tell just by looking at the guinea pig. Now, if we had a hairless guinea pig, we do know for sure that it's little h, because if it did have a capital letter, even one, that dominating allele would take over and it would have hair. So let's try a monohybrid cross. The root mono means one because it focuses on one trait, in this case, hair. And that means a Punnett square would need to be created like this, with 4 squares. In our cross, we are going to cross 2 heterozygous guinea pigs. So let's look at this in steps. Step 1, first, figure out the genotypes of the parents. Heterozygous means big H, little h. Step 2, place one parent along the top of the Punnett square, outside of the boxes like this, and place the other parent along the left side of the square, outside of the boxes like this. Step 3, cross them. For formatting purposes, we always put the capital letter first. The results that you get in the squares would be the offspring, the babies. Now, if I were to ask you the genotypes of the babies, you could list them out. 1 big H, 2 big H, and 1 little h. I could even turn that into a genotype ratio of 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Or a percentage. 25% are homozygous dominant, 50% are heterozygous, and 25% are homozygous recessive. Well, what if I asked about the phenotypes? What does that mean? I like to think that pheno sounds like physical. So the phenotype would be the physical traits of that organism. And in this example, it's whether they have hair or not. Now remember, any babies that have a capital H have a dominating allele, and they will have hair. So the babies that are big H, or big H, little h, they have hair. So 3 of them here, the 1 big H baby and the 2 big H, little h babies, they all have hair. The homozygous recessive baby, which was little h, that baby has no dominant allele present. So this little guy would be hairless. So you could say the phenotype ratio is 3 have hair to 1 that's hairless. Or a phenotype percentage could be 75% hair, 25% hairless. One thing to emphasize about Punnett squares is that they are predictions. These are probabilities. This means it's not necessarily exactly what you're going to get. For example, it's a probability that a child has a 50-50 chance of being born a boy or a girl. But we all know families that only have children that are girls, or families that only have children that are boys. Probabilities are predictions. Another reason to love biology, it's exciting. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Alright, so again, a monohybrid cross technically is a cross between 2 true breeding parents that differ only in one characteristic being studied. So in our case, in the case of the guinea pigs, we can't really apply that because true breeding varieties of guinea pigs don't necessarily exist. But we could do that with our pea plants. So we could take our true breeding variety of yellow seed pea plants and cross it with our true breeding variety of green pea plants. And then we would create a monohybrid. So this monohybrid is only produces yellow seeds. Okay, so this would be 100% yellow seeds. Then again, we allow this to self-fertilize. And in self-fertilizing, we could set up a Punnett square to predict the possible offspring. So a Punnett square is that kind of visual representation of a cross between 2 individuals in which the gametes of each individual is denoted along the top and the sides of the grid, and then we get kind of a probability of what the possible zygotes will be in the center of the grid. So Again, how do you set that up? You look at the parent's gametes. Okay, so here is our parent. Remember, the parent is a diploid. So here the diploid has, it's a diploid, so it has the chromosomes. It has two genes, or two copies of the same gene, and in this case it has big Y, little y. So when it undergoes meiosis, half of its gametes will have the big Y in it. The other half of its gametes will have the little y in it. So we're separating the gametes, right? So there's a 50% chance that the sperm will have a big Y. The 50% chance that the sperm will have the little y. Okay? The other parent, same thing. 50% chance that the egg will have the big Y. 50% chance that the egg will have a little y. So we're setting up the gametes on the top of the grid for one parent and on the side of the grid for the other parent. And it doesn't matter which parent you place where, because you'll get the same results either way. So you can put mom's gametes on top and dad's gametes on the side, or vice versa. Okay? So in this case, the gametes are the same for both parents. Okay? They're both heterozygous. Okay? So big Y, little y. Big Y, little y. And then you take the big Y, bring it down. So big Y, little y, bring it down, little y, bring big Y across, big Y, little y across, little y. And then here are the offspring inside the boxes. Okay, so this is the probability. This doesn't mean that this is exactly what's going to happen. We're going to have a total of 4 pea plants, 3 of them yellow and 1 of them green. No, this is just a probability, okay? So there's a probability that we'll have about 25% will be homozygous dominant, big Y. 50% chance of heterozygous, 25% chance homozygous recessive, okay? Or a 1 to 2 to 1 ratio, or you can even think of it as a 3 to 1 ratio for the phenotypes. There's a 3 to 1 ratio of yellow to green. All right, so that's how we work with Punnett squares. All right, and you'll get lots of practice doing Punnett squares in both the lecture review worksheet this week as well as the lab this week. All right, moving on. Now, all of this is the product of meiosis, right? So in meiosis, if we review, meiosis produces the gametes. And in the gametes, if we recall, each gamete is genetically unique for a couple of reasons. One would be the crossing over effect that occurs during prophase part 1. But another reason is due to the independent assortment of chromosomes, which we'll talk about in a second. And then because of the gametes, the shuffling of the genes, and the Punnett squares likelihood that there's like a 50% chance that there's going to be a big Y in the gamete, and a 50% chance that there's going to be little y in a gamete. That is all due to another law of genetics called Mendel's law of segregation. So in Mendel's law of segregation, it states that paired unit factors, such as genes, segregate equally into gametes such that offspring have an equal likelihood of inheriting any combination of factors. So during meiosis, the job is to take our homologous chromosomes and separate them, right? Okay, so we're taking our homologous chromosomes, we inherited one from mom and one from her dad, and separate them so that they end up in separate daughter cells. Okay, so that way we only inherit one copy of each gene. one from our mom and one from our dad. We don't want 2 copies from our mom and none from our dad, right? So the law of segregation states that our genes are segregated equally, so that we don't inherit more of our genes from our mom and less from our dad, okay? We want to inherit equally. Okay, so that's what the law of segregation is. And that has to do with meiosis, part 1, when we are separating those homologous chromosomes from each other. So remember, the whole goal of meiosis part 1 is to separate the homologous chromosomes from each other. And then meiosis part 2, we're separating the sister chromatids from each other. Okay, so that way we go from diploid to haploid. Each daughter cell has 1 copy of every gene. Started out with 2 copies, now only has 1 copy in every gene, of every gene. Okay, so we're equally distributing the genetic material. All right, moving on. So we talked about homozygous, recessive, and remember how, like in the Amoeba Sisters video, the first one, when she talks about the bitter taste gene, and she knows what her phenotype is. She knows that she is homozygous recessive because she cannot taste the bitter PTC taste, and that indicates that she's recessive. And she knows that her parents must be heterozygous because they can taste it and they're her parents, so they must have each passed on a recessive allele. But what if her sister can taste, but she can't? Okay, like at the end of the video, it said that, the other one, Petunia, could taste. And I don't think Petunia and Pinky are related, but let's say, Petunia could taste, but, she could taste bitter taste. So that means that she has a dominant allele. But we don't know if she's heterozygous or homozygous dominant. So what happens if we have some sort of individual and they have the dominant phenotype, but we don't know their genotype? How do we figure out their genotype? Well, one way is to do a test cross. A test cross is a cross between a dominant expressing individual with an unknown genotype with a homozygous recessive individual. And that way, if we do that, we can look at the offspring phenotypes and indicate whether the unknown parent is heterozygous or homozygous for that dominant trait. Okay? So for example, let's say that we have some mysterious yellow pea plant, and we don't know if this pea plant is homozygous dominant or heterozygous. Okay, so this pea plant could be big Y, or it could be big Y, little y. We don't know which one it is. Okay, so to figure out which one it is, we're going to cross it with a little y, little y. Okay, so there's 2 possibilities. If we cross it with a little y, and we get only yellow pea plants from the offspring, then we know that it must be homozygous dominant. Okay, if all of the offspring are yellow, then this guy must only give yellow alleles, only must give the dominant allele. Okay, that means that all the offspring are heterozygous. They got a little y from the green parent and a big Y from the yellow guy. The other possibility is if you cross the mystery yellow pea with the green pea, and you start seeing some green pea plants show up in the offspring. if you do see some green pea plants show up, then you know that the yellow pea plant was heterozygous. Okay, because if there's homozygous recessive offspring, they must have gotten the other recessive allele from the yellow pea plant. Okay, so doing a test cross will give us an indication of what the genotype is of the mystery plant. Okay, whether it's homozygous dominant or heterozygous, just by looking at what the offspring ended up being. Okay. Now, could you do that with Petunia? Probably not. I don't think she would want to just randomly mate with people and see what type of offspring she produces. In cases of humans, if you wanted to figure out what you are, you got to go back in time instead of, mate with random people. So you do a pedigree. So you would check your parents and your grandparents and your great-grandparents and look for any homozygous recessive people in your ancestry. Okay? So test crosses, don't do that for humans. Okay? So this is more for plants. Sometimes people do that with, like, if they're into breeding of certain animals. They might do that like with dogs and stuff, but not a human thing. So don't do that at home. Not a good idea. Yeah, don't do that at home, kids. Yeah, you can do that with pea plants. Go right ahead. Okay. All right. So here's a visual connection question. In pea plants, round peas, big R, are dominant to wrinkled pea plants, little r. So you do a test cross between a pea plant with wrinkled peas, little r, and a pea plant of unknown genotype that has round peas. So you end up with 3 plants, all of which have round peas. From this data, can you tell if the parent plant is homozygous dominant or heterozygous? Okay. So basically, you have your mystery plant, and you cross it with little, and you end up with a bunch of round plants, or just 3 round plants, round peas. So can you tell if it's big R, or big R, little r, based on 3 offspring? What do you think? Well, I mean, you did the test cross, and you do have 3 plants that show round peas, but I mean, it's only 3 plants, so the size of the population is really small. So I would say that you can't totally be sure that it's homozygous dominant based on just having 3 offspring, okay? I think your data set is a little too small. If it was 300 plants and they all had round peas, you could definitely say, yes, it's homozygous dominant, or even 30. But 3 is just such a small number that it could just be due to chance. So I would say no, you could not tell if the parent plant is homozygous dominant or heterozygous just from only having 3 offspring, because it could just be random. All right. I think it's just too small of a sample size. If this was 30 instead, then I would say yes. Okay, so no, sample size is too small to give a definitive answer. All right. All right. Moving on. So we talked about the law of dominance, where in a heterozygote, the dominant allele masks the effects of the recessive allele. We also talked about the law of segregation, where the gametes, the genes segregate into separate gametes during meiosis. The last law we're going to talk about is Mendel's law of independent assortment. So we talked about did we talk about law of independent assortment? I can't remember, but in metaphase part 1 and metaphase part 2, remember how this is the second opportunity for genetic variation with our gametes, because during metaphase part 1, the homologous chromosomes, they orient themselves independently of one another. Okay, so we could have, you know, the blue line up all on one side and the red all line up on the other side. Or another possibility, we could have the blue here and the red here and the red here and the blue here. Okay, and we talked about earlier how this is just a gamete that, or excuse me, a cell that has just 2 pairs of homologous chromosomes. We have 23 pairs, so that leads to over 8 million different possible combinations of our homologous chromosomes, their arrangements during middle metaphase. Okay, so our law of independent assortment basically adds to genetic variation. So law of independent assortment states that if we look at the homologous chromosomes, they independently assort themselves. Okay, so they're not going to be, you know, deciding, okay, everybody from dad, you know, go to the left, everybody from mom go to the right, right? They're independently assorting themselves. So that means that the genes that these homologous chromosomes carry are assorting themselves, you know, independently as well. So the genes do not influence each other with regard to the sorting of alleles. So if I have a gene on this homologous chromosome, it's going to assort itself independently from a gene found on a different non-homologous chromosome. So if my eye color gene way over here, it'll sort itself differently than my hair color gene way over here. Okay, so genes do not influence each other with regard to sorting of alleles into gametes. Every possible combination of alleles is equally likely to occur. Okay, so that means that if we're tracking 2 different traits or 2 different characteristics, we can do dihybrid, and since they aren't influenced by, the genes aren't influenced by each other in gamete production, they're not going to be linked together. Okay, so we're going to create something called a dihybrid. So dihybrid is the result of a cross between 2 true breeding parents that express different traits for 2 characteristics. So this is different from a monohybrid, so we're going to look at 2 characteristics instead of 1. And we're assuming that these 2 characteristics are on different homologous pairs, okay, so on different chromosomes. And so we're going to watch another Amoeba Sisters video. This one is on dihybrid Punnett squares. So we'll see how it's set up, and then we'll take a closer look.
Have you ever been working on a Punnett square and you think, man, I really wish there were more than just these 4 squares? Well, if you ever have, today is your lucky day. And if not, well, prepare to have more squares. Because up until this point, we have focused on 1 pair of alleles, hence a monohybrid cross, where mono means 1. In guinea pigs, for example, the trait of having or not having hair is influenced by a pair of alleles. But guinea pigs have more traits than just hair, right? Well, if you perform a cross with 2 pairs of alleles, that's called a dihybrid cross. The root di means 2, so 2 pairs of alleles. One of us has this cat named Moo. Moo is in what the cow says, because he kind of looks like a cow, and we're not that creative with names. Anyway, the thing about Moo is that he's a cat that loves sinks. You get up in the morning and you try to splash water on your face, and oh no, there's a cat in your sink. And you try to brush your teeth, and no, there's a cat in your sink. And you have people over and you're terrified they're going to flip out because there's a cat peeking out of the sink. Now, not all cats like sinks, but Moo does. Around the clock, chances are he's in the sink. We cannot find any information about whether this is a genetic trait. We don't know if Moo's parents liked sinks. But then we realized that Moo is not that weird because there's this website called catsandsinks.com. Seriously, Google it. You're going to find tons of cats and sinks. What if this was a genetic trait? Now, there's no research that we can find that demonstrates this is genetic, and it's probably not. Behaviors like that can be challenging to study. But for practicing dihybrid squares, let's imagine what it would be like if that was a genetic trait. So if this was genetic, hypothetically, cats that did like sinks would have to have a dominant allele, capital S, and cats that did not like sinks would have to have a pair of recessive alleles, lowercase s. Let's also take into account that cats, like guinea pigs in our previous video, typically have hair, but they can also be hairless. Having hair will be represented by the allele big H, and not having hair would require two recessive little h alleles. So let's say we want to cross a cat that is heterozygous for the trait of having hair and also for liking sinks. Heterozygous for both traits would be represented by the genotype big H, big S, little s. Now we want to cross that cat with a hairless cat that does not like sinks. To be hairless, the cat must be little h. All it takes is one dominant allele, a capital H in this case, for it to have hair. And if it does not like sinks, a recessive trait in our example, then it is little s. All it takes is one dominant allele, a capital S, and it would like sinks. So the second cat has a genotype of little h, little s. Remember how it looks like in a regular monohybrid Punnett square? Here's an example of what it would look like if you were crossing a big H cat with a little h cat. Remember how you put the parents on the top and the sides like this? Well, when you are doing this, those alleles on the top and the sides, they represent the alleles that would be in the gametes of the parents. Gametes are sperm cells if male and egg cells if female, and they contain half of the genetic material as the cat's body cells. So it makes sense that if there are two alleles, the letters, in the big H, little h parent, then a gamete would only carry one letter, a big H or a little h. This is known as Mendel's law of segregation. The gametes only carry one allele for a gene. Well, if you had a cat that is big H, big S, little s, then there are four alleles there. Two genes, one involving hair and one involving sinks. So if gametes carry only one allele, a letter, per gene, that means that each gamete is going to have two alleles. In those gametes, with each of them having two alleles, the letters, you have to account for every possible combination. Mendel's law of independent assortment says that those alleles are not linked. That means a cat can have hair and like sinks, or not have hair and like sinks. There is no link with the sink. It rhymes. So let's work out a dihybrid with the parent cross here. Step 1, write the parent cross with your 16 square Punnett square. Step 2, we have to get the gamete combinations from the parents. You know, what we write along the top and the side of the Punnett square. We really like the FOIL method to come up with these gamete combinations, but this is not the only way you can do this. We just like this way. So we're going to show you how to FOIL the first parent. And the first parent is big H, big S, little s. So to do FOIL, we do it like this. First, outside, inside, last. And you get these gamete combinations. Let's place those on the top of the Punnett square like this. Now let's go ahead and FOIL the other parent. The other parent is little h, little s. So we'll do first, outside, inside, last. And you get these gamete combinations. Let's place those on the side of the Punnett square like this. And yes, they're all the same for this parent because notice that was all this parent could contribute as far as alleles. Now remember again, each gamete must have one allele, letter, of each gene. That's why you won't find a gamete with only H's or only S's. Remember, one allele of each. And now for step 3, combine the gametes to see what the offspring prediction will be. Now, for formatting purposes, because the parents had H's coming before S's, we're going to write it that way with the offspring as well. For formatting, you should also put capitals of each letter type before the lowercase. So in our example, what is the genotype ratio in the predicted offspring? Now remember that genotypes are the genetic makeups, the letters that represent the alleles. So 4 out of 16 ended up working for all these genotypes. That's a 1 to 1 to 1 to 1 ratio. It's kind of fun to say. Now, what about phenotypes? Well, half of the cats here have hair, and half of the cats here don't. But with dihybrids, often you are asked about both traits. For example, what is the chance that there would be a kitten that has the same phenotype as our Moo? Moo has hair, and he likes sinks. Well, it's a 4 out of 16, 25% chance, that a kitten would have the same phenotype as Moo. We could write out that 4 out of 16 cats have hair and like sinks. 4 out of 16 cats have hair and dislike sinks. 4 out of 16 cats are hairless and like sinks. And 4 out of 16 cats are hairless and dislike sinks. This is also a 1 to 1 to 1 to 1 ratio. Now, some big things to remember. In our example, the genotype and phenotype ratios happen to be the same. This does not always happen. The handout will give you an example of when it's not. Just remember the steps to setting up the problem correctly, and you'll be fine. Also remember that Punnett squares are predictions. This Punnett square is only predicting the chances of having offspring with certain genotypes or phenotypes. It's fascinating, really. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so again, that Amoeba Sisters video, I don't know what was going on with the sound in the background, but anyway, so that gave us a good introduction into how to set up a dihybrid cross and then how to solve one. So we're going to try to solve one with this visual connection question. So in pea plants, going back to Mendel, we have a round seed shape. Big R is dominant to wrinkled seed shape, little r. And yellow peas, big Y, are dominant to green peas, little y. So what are the possible genotypes and phenotypes for a cross between big R, big Y, and little r, big Y, little y, okay? So we need to set up a Punnett square. So first off, it says here, how many squares do you need to do a Punnett square? So let's look at the possible gametes first, okay? So if we set up the gametes, so we have big R, little r, big Y, big Y. So in order to figure out our gametes, we need to do something called FOIL. FOIL stands for first, outer, inner, last. So first means the first of the gametes, okay? So basically big R and big Y, so that's the first. So that's one possible gamete combination. Outer, big R, big Y, so it's these guys. That's another possible gamete combination. Inner, to be these two, little r, the big Y, and then last, these guys, little r, big Y. Now notice from this we have basically big R, big Y, big R, big Y, little r, big Y, and little r, big Y. So basically we have a total of 2 types of gametes. We have big R, big Y, and little r, little y. So I only need to write 2 rows, okay? I don't need to write 4 rows because I'm going to get the same results. Now let's do the other one. So the other one we have little r, and big Y, little y. So I'm going to do FOIL again. So first would be little r, big Y, outer, that's this guy and that guy, so that's little r, little y, inner, little r, big Y, and then last, little r, little y. So again, I only have two different gamete combination. I have little r, big Y, and little r, little y. So I actually only need to set up a Punnett square with these two combinations and these two combinations. So I actually only need to set up a Punnett square with 4. So I'm going to do big R, big Y as one of my gamete combinations, and little r, big Y as the other one from this parent. Okay, you could do all 4, but it's going to give you the same results no matter what. Okay, over here I have, half my gametes are going to have little r, big Y's. Half my gametes will have little r, little y's. Little r, big Y, little r, little y's. Okay, so now I'm going to do the same thing where I'm going to bring these gametes down and these gametes across. However, I like to keep my gametes together. Okay, so I'm going to put the r's together and the y's together. So I'm going to do this a little differently from what I've been doing. So I'm going to combine these two together into this square. So I'm going to put the big R and little r together and the big Y together. Big R, little r together. Big Y, little y together. Little r together. Big Y together. Little r together. Big Y, little y together. Okay, so here's my results, okay, from what we got. Now, if you ended up writing all 4 of these down here and all 4 of these over here, you would have gotten the same results, just double the amount, but we still would have gotten the same ratio. So in this case, we have, we look at the results here. So we have, how many squares do we need? We only needed 4. And as far as the genotypes, we have big R, little r, big Y as one genotype. We have big R, little r, big Y, little y. And then we have little r, big Y. And then finally we have little r, big Y, little y. So those are the 4 genotypes. As far as the phenotypes, we can look at what that means. So big R, little r, so that means round. Okay, so this is round. And then yellow. So both of these are round and yellow. So round yellow. Round yellow. And then this one is wrinkled yellow. These guys down here are wrinkled yellow. Wrinkled. So my genotype is a 1 to 1 to 1 to 1 of these different genotypes. But my phenotype ratio is a 1 round yellow to 1 wrinkled yellow. Okay, so they're all yellowed, but they're so all of them are going to be yellow in coloration, but there's 50% are going to be round, 50% are going to be wrinkled. Okay, So that's my Punnett square. So just realize that when you set up the Punnett square, if you notice that you have duplicates of the genotypes, okay, you don't have to write out all the genotypes of the gametes, okay? Because it's all probability. So if I look here, I have, you know, 2 of my sets of gametes are the same. But remember, gametes, this is just probability. So if I look at this, you know, 50% of my gametes are big R, little, big Y. So I only need to do, you know, 50% of the gametes. 50% are little R, big Y. So I only need two rows there. I don't need 4 rows with 2 of the same. It doesn't make sense. Okay? All right. Let's do some review questions. The observable traits expressed by an organism are described as its. Is it a phenotype, a genotype, the alleles, or a zygote? The observable traits expressed by an organism. If you chose the phenotype, I would agree with you. So the phenotype, physical expression of the trait. Okay. The next question, a recessive trait will be observed in individuals that are blank for that trait. Would it be heterozygous, homozygous or heterozygous, homozygous or diploid? So in order for you to be observed recessive, you have to be homozygous for that, okay? So homozygous is the correct answer. Okay. If you're heterozygous, you show the dominant allele. So homozygous, 2 recessive alleles. C is the correct answer. What are the types of gametes that can be produced by an individual with the genotype A, big A, big B, little b? Okay. So remember FOIL. First, outer, inner, last. So is that first, outer, inner, last? That would be C. And then finally, what is the reason for doing a test cross? Is it A, to identify heterozygous individuals with a dominant phenotype? Is it B, to determine which allele is dominant and which is recessive? Is it C, to identify homozygous recessive individuals in the F2? Or is it D, to determine if 2 genes assort independently? So why do we do a test cross? If you chose A, I would agree with you. So it's to figure out what the genotype is for those with the dominant phenotype. So to identify if it's heterozygous or homozygous dominant. A. Okay. All right, so we are done with 7.2 laws of inheritance. So by now you should be able to explain the relationship between genotypes and phenotypes in dominant and recessive gene systems. You should also be able to use a Punnett square to calculate the expected proportions of genotypes and phenotypes in a monohybrid cross. And then explain Mendel's law of segregation and independent assortment in terms of genetics in the events of meiosis. And then finally explain the purpose and methods of a test cross. So for the final video, we're going to get into 7.3, the extensions of the laws of inheritance. So this is going to be a little bit more complicated because we're going to deal with things like multiple alleles and codominance and that polygenic inheritance that was alluded to earlier and things like that. Okay, so we'll get into that in the next video.